Hello, everyone, and welcome to a new lecture for CPH 290. Today, it's a great pleasure to have a team of speakers from Dandelion Health. We have Luke Sagers and Kiko Wemmer. So Luke is a data scientist at Dandelion Health, where he works on abstracting clinical entities from unstructured text and imaging data, and researches the impact on AI models on clinical care. He recently received his PhD from Harvard University's Department of Biomedical Informatics, and his research there focused on applying synthetically generated imaging data in clinical computer vision pipelines and predictive modeling using biomarkers and EHR data. Kiko is a general manager at Dandelion. She joined the company as the first hire. That's exciting. Foregoing the foundational partnership with Sharp Healthcare and Stanford Health, and establishing Dandelion's operation infrastructure and initial team. Kiko spearheaded the development and launch of Dandelion's AI validation service and the GLP-1 data library. And she now focuses on cultivating an ecosystem of startups, researchers, and pharmaceutical companies to drive equitable, transformative AI innovation in healthcare and life sciences. And before Dandelion, Kiko worked in international development, designing programs and securing scholarships to support underserved women and girls from emerging economies. So thank you so much, Kiko and Luke, for joining us. Very excited about having you talk about Dandelion today, and like maybe you can also talk about what Dandelion does, and like it's a very unique type of company. So I'd love to hear more of you about that. So hand it over to you.
Thank you so much, Ahmed. We're also very excited to be here. We can't see anybody's faces, but maybe that's good. If you're sleeping, we won't see it. But we're Dandelion Health, and thank you for the opportunity to present. Dandelion Health is a real-world data and clinical AI platform for precision medicine and personalized care. Our whole purpose for existence is to help researchers like you, AI startups, maybe the ones that you will found and lead someday, medical device and life sciences companies to accelerate their research and product development for the betterment of patient outcomes. We do this by partnering with diverse health systems across the United States to address the data bottleneck problem in clinical AI research and development. The presentation. So for today, we'll provide an overview of the data bottleneck in healthcare AI research and development. I'm sure it's something that you as researchers are painfully familiar with. And we'll just put the Dandelion point of view on that. And then we'll share a bit about how Dandelion is working to address the data bottleneck challenge. After my brief introduction of that, I will hand it over to my colleague Luke to share an example of how Dandelion's platform, real-world data and AI, was used to do some really interesting research in the cardiometabolic space. I had an intro slide, but we don't need it because Ahmed was an excellent MC, so we'll just go over to the content. Last year, I had the pleasure of going to an academic conference in Cambridge, Massachusetts, and I was listening to a lot of talks by your peers, PhDs and postdocs, on research they were doing in healthcare AI ML. And the number of people who did their research on the MIMIC database blew my mind. It was funny to me because I was also at the time working with one of the creators of the MIMIC database on Dandelion's AI validation tool. And like what he told me was MIMIC is literally like a little hard drive that kind of sits in his closet. So to see that how technology scales, like a little thing that he did over beers with his friend and sits in his closet and he kind of updates it from time to time, and how many papers and ideas and innovations were built. A lot of like early AI solutions also leveraged public data sets like Mimics. I was just like, wow, I really feel lucky to be at a company like Dandelion who is solving a problem like that. Like with data, you can do so much. How can we get more data, better data to unleash wholly new types of questions and projects and solutions for patients? When I was joining Dandelion, this was what the status quo looked like. And it's maybe a little bit about like how your life is today. First, you come up with ideas based on the research or based on the data you think you can get, instead of starting necessarily with first principles about what are the questions worth asking, what are the problems worth solving, because we have to be practical. The research has to get done and we need data to do it. And so after coming up with an idea based on data we think we can get, then individual companies, individual researchers would go to individual hospital systems or even individual people at hospitals to beg, borrow, and just try to do whatever they can to try to get some data. And the transaction costs were really high. It was like every individual researcher is kind of bearing the same cost of time and effort to get the data. Individual companies are bearing the cost of the contracting, the cleaning, the preparation to use any bit of data. And whatever data they're able to get tended to be narrow. It was really small, just what was doable within a small agreement. It wasn't necessarily linked to outcomes. Wasn't usually a multimodal database leveraging, high dimensional data assets. And so as a whole, there was just like innovation that could be happening wasn't happening. And it wasn't because the innovation was impossible or we didn't have the tools. Like the data was there, the compute was becoming cheaper. There were really smart people wanting to work on these projects. It was just about risk aversion and not like about sharing data, and then also capabilities of de-identifying data at scale and making it available. And so Dandelion exists really as kind of a utility. You think about all of the companies and all of the products that have been able to be built off of like cloud. Dandelion is likewise a utility for data. Imagine all that could be built if you just had this amazing data asset and it was easy to use, accessible, clean, high quality. And so compared to the status quo, Dandelion's platform is dynamic and meant to address a lot of that. So we're a two-sided platform. On the one side, we have our health system partners, our core consortium. The partners we've announced so far are Sharp Healthcare in Southern California, Sanford Health, which is the largest rural health system in the United States, and Texas Health Resources in the South. We're announcing a fourth and a fifth very soon. And in our agreements with them, we have access to all of their clinical data. We clean it, we structure it, we make it so easy to use for research on day one, and we make that data available to our partners in academia and in industry. The key features that make the data Dandelion is preparing really great for research is a few things. One is that it's multimodal. We're going beyond structured claims, structured EHR, and we're pulling in high dimensional data linked to ground truth outcomes. It is longitudinal and it is representative. We have representation in terms of care practice diversity, machine diversity, patient demographic diversity, and that's why we select our partners to be community hospital systems for the first three, and one in each major region of the United States. Yeah, I'll just move on for there. This is just to underscore the point about the data being representative. So no one health system is going to represent, all of the United States, even on one dimension, such as race and ethnicity. And so on the left, what we have is in the gray that's the US Census average. And each health system as a consortium actually does quite well, so that for research purposes or product development, you could even upsample on machines or patient subgroups that are underrepresented in existing studies or are meaningful in some way for the research or product you're trying to develop. The data come from all across the integrated delivery network. So this is everything that's happening in inpatient, outpatient, and tertiary care. And the health systems we partner with are leaders in a number of clinical areas that are important, such as cardiology, cancer, women's health, neuroscience. A lot of treatments are being developed in these areas today, and these health systems really sit at the forefront of capturing the data that are going to make that innovation possible. The data, it cannot be understated how important it is to use multimodal data, but also how hard it can be. And that's why Dandelion exists. So we go into a single health system like Sharp, and we find all the IT systems that exist in that health system. We have a team, powered by the likes of Luke, doctors, pharmacists, clinical informaticists who go in, they de-identify that data, they clean it up, they structure it, and then they harmonize it across the health systems. And then in the end, you're able to have really rich clinical information for a number of disease areas across therapeutic areas. And Luke is going to share an example of how we do that. The catch sometimes with using multimodal high dimensional data, such as imaging and waveforms and notes, is that not everybody, not every team has a PhD in computer vision or natural language processing. And so when we're making this data available to researchers, we also want to make the multimodal data easy to use on day one. And so in that sense, we have an AI part of our company, which is validated by CHAI. So CHAI is the Consortium for Health AI. They have quality assurance labs that allow people who have existing algorithms to run it on the data in the quality assurance lab to ensure that the algorithm is performing as the developer says it does, but that it's also performing fairly across subgroups of interest. Dandelion is one of the quality assurance labs, and we have a platform that allows algorithms to run on our data as an external holdout set, and then we output a performance report. We've actually been able to help AI startups use that performance report to support their regulatory submission. But then we also create kind of a deployment arm for them, because if there is a use case in the market that requires imaging or waveforms or notes, for example, and there's a really high quality algorithm, for example, that segments and quantifies what's in an abdominal CT, we can then pair those two together and output labels that are easy for quick analysis. And then, the most important thing is that ground truth outcomes are linked to all of the data that we are delivering for researchers and industry. When I started Dandelion, ground truth meant, what did the doctor say, or what did 3 doctors say when they adjudicated some outcome? And that is one way to get at ground truth, but there's a lot of truth existing in the pathology slide, like what's actually happening at the level of cells and tissue and organs. And so the real sweet opportunity here is to go beyond ICD-10 codes and what was in the structured information and start to use the imaging, the waveforms, the electronic health records, lab values, and vitals to start to triangulate what's actually happening at the level of disease, and then do really interesting things like phenotyping patients, having better quantifiable endpoints for research, and so on. Here is the summary. So one leg of the stool, real world data of a granularity that is difficult to access today, but Dandelion makes it easier to access it. We have the clinical AI platform that is used to develop and validate algorithms just on their own as products, but also where there's a use case to run the algorithm on the real world data to extract structured outputs for faster research. And then the third leg of the stool are people like you with the ideas, the use cases, and the talent who want to leverage these capabilities to build things that don't exist today. With that, I'm going to hand it over to Luke to provide an example of how Dandelion did something interesting in the real world.
Thanks so much, Kiko. It's great to be here. It's great to have this opportunity to share some interesting research that Dandelion has done. And what I tried to do is choose a study that highlights several of Dandelion's capabilities, so you can kind of get an idea of really what this data can unlock in kind of an end-to-end research pipeline and the type of insights that can be generated using Dandelion data. You can go ahead to the next slide. So by now, I'm sure many or most of you are familiar with these GLP-1 receptor agonist drugs, these weight loss drugs that were originally used for management of diabetes and have since been labeled and approved for weight loss themselves. So around August of last year, there was the results came out from a clinical trial that was later published in the New England Journal of Medicine, studying the effects of semaglutide, one of these GLP-RA drugs, and looking at major cardiovascular events. You can pan forward one. So this study, what it was trying to determine was whether these drugs have cardioprotective benefits or whether they can reduce major adverse cardiac events like myocardial infarction and stroke in a subset of patients who did not have diabetes. Like I mentioned previously, this drug was approved mainly for the management of diabetes. And so this trial went a step further to say, hey, we want to look at people who don't have diabetes, but who do have a condition, a pre-existing cardiovascular condition and so this study looked at severe pre-existing cardiovascular disease patients who had BMI above 27, but without diabetes. And what they found was very interesting, pretty stunning, actually, in the pharmaceutical world. Was they saw a 20% reduction in these major adverse cardiac events, and that was over a mean follow-up time around 40 months. And this facilitated the approval for semaglutide to be prescribed to reduce these cardiac events in this population, the population who has existing and pre-existing cardiovascular disease. You can go ahead one more. Now, we love clinical trials because they are the gold standard for evidence generation. And so, of course, real world data at this point isn't ready to replace clinical trials. However, clinical trials obviously have severe limitations as well. They have strict inclusion and exclusion criteria that often limits their diversity. So in the case of this study, 85% of the study cohort was white, and of that, I think it was an additional 73% was male. So the white male overrepresentation in clinical trials has been a long-standing problem and continues to exist, as this was completed only last year. Clinical trials are very time-consuming. You have to enroll enough patients, and in the case of major adverse cardiac events, you have to be able to collect enough of those events in order to see, the signal that you're looking for. And so this can take years and is very expensive. Every time you update the trial protocol, every time you have patients drop out, which in this case, thousands of patients did drop out of the study, it's a very time-consuming and expensive process, of course. And so we were motivated by this study to examine in the real-world data, is there a way that we could build upon or expand this cohort population to generalize some of these findings to a more general population that does not have pre-existing cardiovascular disease? So you can go ahead, another slide. So the research questions that we wanted to answer was, is there a potential for GLP-1 RA drugs to benefit patients in primary prevention, like I mentioned, that trial looks at those who have cardiovascular disease. What about for those who do not have cardiovascular disease? Is there cardioprotective benefit that is being unstudied by the SELECT trial? We wanted to know if we could use real world data and AI to avoid some of the obstacles posed by RCTs. Can we do this faster? Do we have the capability to expand and contract populations and look at diverse population groups across time? And then we also wanted to know, and with an AI partner, an AI developer partner, can we leverage AI to predict these events, but not only to predict without having to wait for these events to occur? So essentially, can we use artificial intelligence and biological signal to predict MACE risk using unstructured data and use that to see if we can identify any cardioprotective benefits in these more generalized populations? Go ahead, one more. So we partnered with a company out of Germany called Fieron. They are an clinical AI developer, and they have an algorithm that they have trained on internal data that predicts MACE risk from 12 lead ECGs. So MACE risk, there's many prediction algorithms that are out there, and many of them take in a lot of different modalities, clinical EHR data, and the sort. This one was a little bit unique because as is, I think there's more of these popping up, but it looks just directly at the 12 lead ECG, so waveform data, which is able to capture biological signal. This is unstructured, and so because it's unstructured, our hope and our intuition is that it will be able to both be unbiased by clinically administrative labels that come from clinical visits or from limitations to access to care or the type of visit that you're receiving that records in the HR, but also that because it's detecting these nuanced and subtle biological signals, we may be able to detect significant differences in MACE risk before having to have the patient experience a major adverse cardiac event. So we might be able to see distinguishable differences in patients who in a clinical trial would both be considered, pre-event, so in the healthy population, I guess, but they still might have different cardiovascular profiles that could be identified using unstructured data like this. Go ahead. So this is kind of the exciting part where Dandelions gets to shine, is that before we tried to study the question of whether GLP-1s are, cardioprotective in a greater population, we wanted to validate Fieron's algorithm using the validation service that Kiko mentioned. And so what we were able to do, because we have access to all this diverse longitudinal data, we were able to randomly select 100,000 ECGs for a validation test on the algorithm before we use it in our study. And so we chose 100,000 ECGs with the clinical outcomes of myocardial infarction and stroke, which were the major events noted by the trial. And we wanted to validate Fieron's algorithm to see, how well does it predict these events in our data set, which we have curated, which is diverse. So in this data set, I think of 100,000, 38,000 were white. So a lot more diversity across racial and ethnic groups. We were equally divided by sex, and we also had measures of rurality and social vulnerability index and median decile income decile. So we had a lot of this information that would allow us to both see how the algorithm works overall, but also see if there are any systemic biases that we should be aware of before deploying an algorithm like this in practice. Go ahead. So what we found is that their algorithm actually performed quite well. So on what to them is a completely external data set, the AUROC for predicting myocardial infarction was 0.81. And for stroke, it was 0.75. These are, I think, very respectable and very robust AUCs, especially for a completely external data set. You look at some of the most popular risk scores in the field, things like the Framingham risk score and the Prevent calculator, and they are, somewhere in that 70 to 75, maybe 77 range. So this is really exciting and robust validation for an external data set. And as you can see in these ROC curves, we also were able to, and like I mentioned before, validate this algorithm across different subgroups. So these ROC curves are plotted with the color being race ethnicity group. And as you can see, there's actually not a ton of variation. And this algorithm was performed fairly normally, fairly equally across these racial ethnic groups, as well as across income groups, across sex, and all of the groups we studied. So essentially, we were able to validate and vet their algorithm before applying it to data, which was really important for our study to have more confidence in our study results. And we could do that using internal Dandelion data. Okay, go ahead to the next slide. So the next step was we were going to define a cohort. Again, we have a lot of data that we have access to. So top line, we started to pare down eligible patients using the same trial protocols that they used in the SELECT trial. And so with one exception. So like I mentioned, instead of just studying people who had pre-existing cardiovascular disease, we dropped that requirement and looked at those who were obese or overweight, because those are still important labels for the drug prescription. So those who are obese and overweight, but who did not have to have pre-existing cardiovascular disease. And what we were able to do was we were able to get a finalized cohort of individuals who were taking GLP-1 RA drugs and those who were not. So our positive cohort down on the bottom left are those who are taking the drugs. Negative cohort are those who are not taking the drugs. And we were able to get we were very able to very quickly create a trial population, a simulated trial population that is powered enough to find biologicals or to find signal in the differences. It's important to also note that we actually had no overlap between the ECG that we used to validate this algorithm and the ECGs that we were going to use in this trial, in this emulated trial study. Okay, you can go to the next one. So yeah, the study itself was fairly simple. So we had this cohort of 20,000 plus individuals with at least one ECG, a few other requirements like multiple endo or PCP visits, so that we had at least some sort of longitudinal data and follow up on these patients. And we fed them to this Fioran algorithm that we had validated, and we were able to generate risk scores, risk scores for the group who was on the GLP-1 drugs, and we were able to compare that with the group who were not on the GLP-1 drugs. So pretty simple design there. You can go to the next one. What we found was pretty interesting. So the modeling was done, I think, using inverse propensity score weighted regression with an interaction term between the drug use and how long the patient has been on the drug. And what we're able to see is that, so in blue, you have this group, this non-GLP-1 group, and in orange, this GLP-1 group. We were able to see that around a year and a half into taking the drug, there was a significant difference in risk that emerged, in risk score as output by the algorithm that emerged in these two populations. And that the risk scores for those who were taking GLP-1s, they were initially on actually a kind of a worse trajectory, but the longer they took the GLP-1 drug, the more their risk score attenuated, whereas those who were not in the GLP-1 group, their risk score continued to rise. And so we did this for both myocardial infarction. This is this chart, and then you can go on forward. The stroke risk score incident stroke actually looks almost identical. So we saw this attenuation of risk in this population who took the GLP-1 drugs without having to follow up and wait for this population to actually have these cardiac events that would be, devastating to their health. You can go one further. So in summary, what we were able to do was we were able to attenuate or we were able to see that the risk for major adverse cardiac events was actually 15 to 20% lower between those who were on the GLP-1 group and not on the GLP-1 group after 3 years into being prescribed this drug. The time to effect, we were able to observe statistically, as I mentioned, statistically significant differences that were visible after 1.7 years. This is much less time than it took them to do this in the SELECT clinical trial. And then the purpose of our study, like I mentioned earlier, was to see if we could expand the population, and in this way see what the potential protective effects are of GLP-1 drugs on a population that did not previously have cardiovascular disease or severe cardiovascular disease. And in this instance, if you kind of do the math and extrapolate from our population size to the population of the United States, this would increase the the group that was able to take these medications from, I don't know how many the original number will, but increase it by an additional 44 million patients. And this isn't to say that, like, oh, we did this study, now go prescribe GLP-1s to these 44 million additional patients because it shows that it can have cardio protective benefits for all of them. But what this is showing is that there is potentially a signal there that should be investigated. And what it could do is it can de-risk a clinical trial that can then look at, a broader subgroup and say, you know what, this real world data plus AI study has shown us that the signal exists. Now we can be much more confident in initiating a multi-year, multi-million-dollar clinical trial studying something similar. And the other thing that we can do is we could do the same thing but break it into different subgroups, and we can look at, for example, Hispanic patients or those with a BMI under 30 but over 25, or those who were in the the most elderly population, the 65 to 80 group. And in our study, we showed that those patients have the greatest risk reduction. That type of result can inform clinical care as well, and it can inform future clinical trials. Again, one of the most exciting parts about all of this is that we were able to get the ECGs that we needed, validate Fieron's algorithm, and complete this whole analysis within just 6 weeks. And so instead of having to wait months and years, or instead of having to, like Kiko mentioned earlier, wrestle with data providers for the right types of real world data that would be sufficient for our our diversity needs, for our representation needs, and for our sample size, we were able to find this population, this cohort, within data that already exists. And we were able to do that quickly and effectively within just a few weeks. And then the fun thing too is that because we have the data, is that future research in this area, or different subgroups, become very accessible and very quickly iterable. So if let's say we wanted to just look at this group of Hispanic patients with a BMI between 25 and 30 or 35, we could just pare down our patient population and rerun the algorithm again, and have updated results, which is really nice. And we can do this for an essentially unlimited number of questions because the data exists in a clean form, a queryable form, and because we because of all the work that we put in on the front end to get the data into that form. So you can go ahead one more. I think I briefly mentioned most of these. So again, this isn't to say that this real world observational data is a replacement for clinical trials. It's not, and we don't have the same claims that you can make from a well-controlled clinical trial. That's still the gold standard. But like I mentioned, these types of studies are clinically informative. They can be used to de-risk clinical trials, but they can also be used to inform clinical practice. There's during my PhD program, I worked a lot with the NHANES data, National Health and Examination Survey data, and that's an observational study as well. They are very rigorous in their sampling, and they do a good job in trying to get a cross section of the United States. But there are actually many clinical guidelines that are informed by NHANES observational study data and not strictly by clinical trial data. So this data is valuable, and these studies are valuable in helping to examine, for example, the effects of interventions before endpoints occur. And in our case, the other exciting part of this was that, like I mentioned before, the ECG is biological signal. It's unstructured biological signal, and because we validated its predictive power on an independent data set, we could have at least a decent degree of confidence that the signal that it's picking up on is real. And when those signal pathways start to diverge, we could potentially intervene, or you could imagine a primary care physician intervening to say, hey, you know what, your risk score is on a bad trajectory. It's not on a good track. Instead of waiting until that patient gets to the point where their symptoms are severe, or worst case scenario, the major adverse cardiac event actually occurs. And the clean data, like I already said this before, it just accelerates everything. Go ahead, Gigo. So, okay, this is just, I'm going to briefly touch on one more study that is with a different, totally different health, or I guess partner. So this is a study that University of Wisconsin at Madison is looking into. So when we talk about GLP-1 weight loss, there's been all these trials and these studies showing that the weight loss is happening, and so it is effective for weight loss. But from a clinical perspective, the type of weight loss is actually very important. It's not so it's not such a good thing if your patient is losing muscle mass or bone density, as opposed to different types of fat. And so you can advance the slide. And so we have a group of researchers at the University of Wisconsin-Madison who are trying to track body composition changes over time using imaging data. And so you can go one more forward, actually. It's probably better to, or actually one more, probably better to have a visual of it. So what they're able to do, what they're able to do is to take abdominal CTs, and they have an AI algorithm they've trained to segment and identify different types, I guess, different substances within the CT. So differentiate between visceral fat, subcutaneous fat, muscle area, and liver volume. And using this unstructured imaging data, they're able to apply their algorithm and use the segmentation to get measured values. How much visceral fat has the patient lost? How much muscle area have they lost? And you can convert that into muscle mass loss. What's their liver volume, and how are these drugs affecting the liver? And again, it's the power of having raw unstructured data to get biological signals, and then link that to their GLP-1 history, to their other clinical histories and studies, and to their patient notes and the abstraction of clinical and physician comments that are found just in the physician notes. And that is able to provide a more full picture of health and of wellness. And we're really excited about the many opportunities for researchers to apply their great ideas to this data that we've been working so hard to curate. And because like these are just two examples of an unlimited number of things that you could do with this data, and we're just trying to build a resource that will allow researchers to do research to validate algorithms and to do all of this in a way that is robust, in a way that includes cross sections of the at least the United States at this point population who are underrepresented traditionally in this type of research. And so it's a very exciting thing in my opinion, which is why I joined Dandelion a few months ago. You can go ahead, another one. I want to spend maybe just like a few minutes in case this is helpful for anybody about my experience going from my PhD, which I just graduated in May, to an industry position. You can go forward one more. So I was in my PhD was lasted 4 years. The first 3 I felt like I was just going to be there forever, and then the last one snuck up on me. And like you have to finish your dissertation, and you want to be prepared and looking for a job, and everything seemed to happen so fast. And so if from my experience, something that really helped me was along the way, starting in year 1, or I guess starting wherever you are, whenever I had presentations like this, or lunch and learns or seminars or things like that, I would kind of just jot down names, notes, and email addresses, and just if I thought it was interesting and like it might be a cool place to work. And I did that with Dandelion actually a few years before joining. I did that with another company, Viz.ai, a few other companies. And with all the companies I did that to, even though I reached out to people like maybe a year or 2 later, they were all happy to facilitate introductions. And they were all like, I interviewed with all the companies who I thought were cool at the time, it's awesome. So keep track of those. Maybe explore an internship along the way. Reach out early. This is another thing that I thought was interesting. So in my job search, all of the positions that I interviewed for, none of them actually existed out on the market yet. So it was because I reached out to the people who I had talked to or who came to present at the companies. I reached out to them. They're like, hey, we might have a position opening in 6 months, but it's not live yet. I can get you talking to the right people. And so actually with all 4 of my interviews the companies that I was interviewing with, none of them were on LinkedIn or Indeed. They were all pre-release interviews. So just something that I thought was interesting, maybe worth sharing. And then of course you can leverage your skills that you learned in the PhD to make a positive impact in the field. I think of those skills, probably the greatest one that you're learning is how to be on the fly, be scrappy, to learn quickly, and to ingest like what's the latest happening in the field and try to push that out into actionable research, into good questions, into thoughtful analyses, and in some cases pushback about research and development. You can go forward one more. And then this is just a brief day in what I do here in case anybody's interested in Dandelion. As a data scientist, I wear many hats. I work with many data modalities, and I try to solve a lot of problems that just seem to keep popping up with the issue of cleaning data. Like Gico mentioned, it's a huge undertaking to ingest data from 3 disparate health systems who are all doing things very differently and try to harmonize those at scale. There's all kinds of weird stuff that pops up. There's all kinds of crazy problems, fires to put out. But it's really fun and it's very rewarding. And when you see comments like, I think there was a comment in the Slack today from an industry partner who said, hey, we've never seen echo measurement data as clean as what you've presented to us. And like that's just really rewarding when you hear from a group that's been working with, major health care organizations for years and years, that they've never seen data that is this clean and this organized before. And so it's very rewarding. I do a lot with clinical notes and right now Echo Video. So abstracting unstructured data or abstracting structured data from the unstructured clinical note, trying to leverage NLP, rules-based approaches, anything we can to get clinical entities out of these notes. And then, yeah, building hopefully what are useful and valuable data sets for clients so that they can pursue primary research opportunities. I'm sorry, I kind of speak fast. I apologize. I'll take a breath. I'm going to turn it back over to you all and to Kiko as well. In case you have any questions, we can open up for a Q&A. And it can be, just about anything you've heard today about Dandelion or some of the research that we've done. You can take it easy on me on all the technical details. Most of this work was done before I joined, but I'll do my best. So thank you so much for having us.
Silent claps, but I see your claps. Does anybody have questions?
Yeah,
Or maybe they're in the chat.
I think they're muted.
Oh, okay. I can like MC and then
Okay.
we can decide. Any chance you've looked at PAD subgroup and an outcome of male as well, assuming a highly diabetic population? Would be very interested in seeing those results. From Colleen Flanagan.
Okay, thank you for this question. This is not a group that we have looked into, and the main, I guess, it's not something that we thought of, but that's kind of the reason why we love pairing with researchers, because everybody has things that they're like, oh, if you would have checked this out, maybe you could have seen this signal, or here's an opportunity. And that's exactly what we're hoping for. And so this isn't a study that we've done, but something that could probably be very interesting. And yeah, we're open to looking for whether we have data on that for sure.
Yeah, we can take questions from the room. Sorry, we were clapping, but I think we were muted as well. So we have one question.
I'm curious, do the clients actually, do they own the data? Like, for example, could a researcher, after publishing some findings, open source some of their data? Or is it like, who owns the data actually?
Good question. Ultimately, the owners of the data are the hospital systems that are collecting it and are the stewards of patient care. So what we do is we license access to curated, cleaned data sets. And that's how it works, but no downloading, no owning, all that belongs to the hospitals.
Oh, sorry. Just to clarify, like, so for example, say the hospital system says, yes, you could open source some subset of the data. Could the researcher do that upon time of like presenting the findings?
We've never asked the question because we've never been requested to do that. So to date, it hasn't happened, but it's definitely something we could ask about. Good question, though. Any other questions?
Yeah, I had a question about clinical notes. You seem to have looked into ECGs and like abdominal CT scans, but are you also able, because I think clinical notes are more susceptible to re-identification. So I'm not sure, when you work with all of these health systems, do you have clinical notes in the first place? And like, if you do you have like in-house the identification kind of process, or do you just work with the different identification processes at the hospital system end?
That's a really good question.
Yeah.
Yeah, this is something that so short answer right now, the notes that I work with for clinical abstraction, I work with inside an environment that is on-prem or I guess I work remote, but it's within the health system. And the reason why is the same reason. It's the de-identification risk. We're in the process of de-identifying notes at scale so that they can come into our environment. And it is it is a difficult thing to do. In the meantime, what they are allowing us to do and what I do pretty consistently is I will run abstraction. I mean, it's a full, I run it, it's a full AWS environment on the health system side. And so I'll run abstraction on the clinical notes, which there are so many clinical notes. And then I will have to take whatever I abstract and kind of do a QA with a subject matter expert at the health system. And then we can extract those labels into our data. So we're starting to get abstracted note data into our system, but we don't have the notes yet at this point.
Okay. And just another like related question. Are you picking the health systems so that you are able to, like, one problem with analysis of real world data in general is that patients might go from one health system to the other. Like when you're collecting this longitudinal data, maybe a patient was admitted to one hospital and then they discharged, but then they get like primary care somewhere else. Are you taking this into consideration when you're kind of working with these multiple health systems to build like a coherent cohort, or is this like a problem that's still hard to solve even though we have like many resources of patient data?
Yeah, that's a great question. I can take this one.
Yeah.
When we were selecting our health system partners, we were very intentional about this. Yes, how do we capture longitudinality, basically? And the way we address it is two things. One, in the selection of the health systems, and the second is through Datavant tokens. So in the selection of the health systems, we partnered with systems who have good longitudinality because either they are the only hospital system in town, like they're the monopolist or duopolist in their local healthcare market. And so patients just go there, and if they move away, and then we try to find them via Datavant token. But for the most part, if you don't move, you go to these health systems and the data are captured there. And in our agreements with the health systems, we get access to all their clinical data, inpatient, outpatient, tertiary, specialty. So we capture all the care that's delivered, assuming the patient stays in the health system. The other thing is that the health systems we chose have insurance setups that also incentivize patients to stay in the health system. So that was a design choice. And then, but it's still real world data. People move, people leave all the time. And so we tokenize each patient at the patient level, and then things that aren't captured well in the health system routine care, we can also get that through token. Things like mortality, not captured systematically by hospitals, prescription fills, things like that. That's really important for research because you want to know if the patient actually took the medication that you're trying to study. Hope that answered your question.
Yeah. There's a question in the chat about data harmonization. I think like what does it take to really harmonize data across these health systems? And I would add like another related question, which is like the validation service you offer. Like what kind of interfacing, like if I build a model in UCSF, for instance, and I want to validate it on your end, like do I need to, is like a standardized format of what the input should look like so that the model actually works in your...
Yeah, good questions. Luke, should we answer the validation one or the harmonization? I think we have 2 minutes.
I'll start with I'll start quickly for the harmonization one. So we have clinical experts that live, clinical informaticists that live in like these mapping spreadsheets that are, massive and extremely in depth. We map across procedure codes. There are some commonalities like ICDs and CPTs, but a lot of it is just, looking at the data, getting into the data, and having our clinical experts derive what are the shared common columns and what are the ones that are more difficult to harmonize. So huge scale mapping, very manually intensive in that regard.
And then on the how it works for validating algorithms, we provide a data menu that has a big section about just tell us about your algorithm, what are the inputs, what are the outputs, and how do the data need to be structured? So like really specific things around the machine settings and the metadata to make sure that the algorithm will actually run on this data. And then typically there is like a small sample of data that they can run their algorithm on to make sure that it works. And then they port their algorithm into our environment via Docker container. And then we do the validation internally and we generate a performance report.
Other last pressing questions in the room?
Sorry, I have a question. I'm curious, so over time, I would assume that Dandelion would eventually have like a very, very large amount of data formatted from like many, many different applications. Do you foresee any kind of future study that could look at all of the data collected or formatted by Dandelion? Like, for example, once you have like a huge corpus, like is the formatting consistent across each application? Do you see any use of like large scale data across many different kinds of applications? That makes sense.
Yeah, what do you mean by applications in this context?
Basically, my understanding is that Dandelion formats the data from the healthcare system for like a company or a researcher, for example, to then use for their specific application. For example, training a model for like maybe solving some medical application. So over time, you would have like a bunch of those. Like you'd have like a huge corpus of data from solving a variety of healthcare related problems. I'm curious if you foresee any future use of all of the formatted data data formatted by Dandelion. Like, is that something you're thinking of?
Yeah, I think so. I think the model is actually a little bit flipped. So there's elements of us creating these data sets, curating for client requests. But what we're actually trying to do is build what you're talking about originally. So we're trying to format all of this data and then let the clients choose from that large corpus that we're actually formatting. So we're building that, I guess, concurrently with each one of these one-off client requests. And yeah, the implications, especially as we get more and more years, are large for actually very multimodal longitudinal studies at scale, and it just becomes more valuable the more time and the more health systems we onboard.
Okay, got it. Yeah, thank you. That helps to clarify.
Okay, thank you so much, Luke and Kiko.
Thank you.
Thank you for having us. Feel free to share my email as well if anyone wants to reach out. I'd be happy to chat.
Thank you so much.
Thanks, everyone. Bye.
See you guys.